Welcome back, my microbiology students. Today we're going to be covering chapter 11, which discusses how humans and microbes interact with one another. An outline for this chapter is as follows. We're going to have 4 sections. The first one will discuss the human microbiome. We'll talk about how colonization leads to disease. We'll talk about how infections are transmitted, and we will tail at the end of this with a little bit of epidemiology, which is the study of how diseases spread in populations. So, first and foremost, let's begin with the human microbiome, which is arguably, to me, one of the most interesting things currently in microbiology research. So, here's a fun fact for you. There are more bacteria cells in and on your body than there are cells that actually belong to you. Those bacteria are called the human microbiome. They are natural, they are important, they are there for various reasons, but they contribute to our health and function of ourselves, of the host organism. So here's a nice infographic that illustrates that point. If we look at this human here, 43% of the cells that are in and on this human are human cells, but the other 57%, the majority of the cells, are actually microbes. So that should go to say that clearly our human microbiome, the natural flora of bacteria that we have in and on our bodies, are probably very, very important to us. And there's a lot of current research that is invested into this exact concept, and it's called the Human Microbiome Project. It is initiated in 2007. And the goal was to characterize various microbes that are living in and on our bodies when we are healthy and determine how they work. What do they do? What purposes do they serve? And how do people who are sick have a microbiome that compares? Can we identify any variations in microbiomes that could potentially lead to disease? And the answer is yes, that it can cause certain risk factors. So, here's some important and surprising results that we found from the Human Microbiome Project to date. This is not an all-inclusive list, but is nonetheless interesting. We have found that human cells, if we were to analyze our human DNA, we have about 21,000 protein encoding genes. Now, we can make many more proteins than just 21,000 due to alternative splicing, and we won't really get into that in this class, but if you took biology with me, you may still remember that. But our microbiota, the members of the bacterial population that live in and on us, have millions of protein encoding genes. Microbes are found in places we previously thought they weren't. For example, in the uterus, brain, the lungs, we thought those areas must be sterile, but they're not. They're just bacteria that are there in small amounts. In fecal matter, there is 100 million viruses per gram, and that is an astronomical amount in such a small specimen. So all healthy people, they harbor these potentially dangerous pathogens, but in low numbers, and that's normal. And the makeup of one's intestinal biota can influence our overall health. So I'm sure now, this has even been on the news this morning, but you'll see commercials and advertisements for probiotics and prebiotics and certain kinds of yogurt to help influence your normal gut flora. And that's because there has been so many research studies that have indicated that it is likely that these intestinal microorganisms influence our overall health. So how did we acquire this microbiota? Where did it come from? So this is again called colonization. When a microbe colonizes our body, think of it like like when I don't know, like Christopher Columbus came over on the sea and colonized America or something to that nature. When we have bacteria that come into our body and take up residence, begin to live in and on our body, that is what colonization is. So how does this happen in a fetus? Again, previously we thought the uterus and all its contents were sterile, meaning that no microorganisms would be present. However, we found later and recently that we were wrong. The placenta actually does harbor a small but significant amount of bacteria of various different kinds that will allow for the colonization of the fetus in utero. During birth, the colonization of the newborn occurs. The movement through the vagina itself, which has bacteria in it, is going to pass on microbes to the baby. So specifically, there's lots of different bacteria of the genus Lactobacillus that will provide the baby with necessary enzymes to digest milk. There's other species that may protect the baby's skin from certain conditions and disorders. And human milk, the act of breastfeeding, it contains around 600 species of bacteria and sugars that otherwise the baby would not be able to digest, but are digested as long as they have a healthy gut bacteria that is capable of digesting it for them. And that hopefully is no surprise to y'all, because we've talked a little bit about the things that microbes eat. Remember, we talked about our microbial nutrition. Some of them eat certain sugars like lactose and glucose, etc. So as long as we have the correct gut flora, we'll be able to digest these components. Now, something that kind of segues us into that subject is C-section. So 30% of babies are born by C-section in the U.S. And so this begs to ask the question if the gut microbiomes of C-section babies are going to be very different from vaginal births. And if that's the case, what are the long-term effects? Are these individuals going to suffer from, let's say, more episodes of colic? Are they going to be more prone to disorders like lactose intolerance, etc.? Maybe not. There is still lots of ongoing research into this, but it is interesting to know that all of this research that is ongoing is so applicable to things that we never even thought were that big of a deal. Our gut microbiome, such tiny things, but wow, how interesting is it that our gut flora is way more important than you would maybe once thought. So in summary, how babies get their microbiome: they get it initially through in the uterus, the act of birth, milk from mothers, caregivers, family and siblings, we share microbes with one another, and their environment. Now, our human body has a variety of environmental niches. What does that mean? A niche is a specific area that has specific characteristics. So, for example, temperature, pH, nutrients, oxygen. Our blood pH is around 7.35 to 7.45, whereas the stomach pH is very close to 0 to 1. So various areas in our body will have variable pH levels, and therefore different microbes will prefer those areas. Same with temperature, same with nutrient and oxygen availability. And so because we have such different variety of niches throughout our entire body, it supports a very wide range of microbes. So lots of different microbes are capable of growing and colonizing and living happily inside and on the human body. Now, this is good, though. This is a good thing. Remember, microbes are good for us in certain ways. Not all of them are pathogens. Now, this gut microbiome and also other bacteria that live in other areas in and on our body produce something called microbial antagonism, and that's where they inhibit other bacteria from growing. So just like any other species, whether it be wolves, bears, things that aren't microbes, they compete in their environments for food and space, etc. Microbes do too. So these normal biota, these normal microbes, are unlikely to be displaced by incoming microbes, meaning those that don't live in there normally in and on our body, because these other microbes are already there and will outcompete them for the stuff that they need nutritionally to exist. There's also chemical or physiological environments within the body that are created by these resident microbes that will inhibit these other hostile microbes from being able to colonize the body. So again, that's just another methodology of microbial antagonism where our normal biota is beneficial to us, or at least, or at worst, commensal, meaning not causing us any problems while they benefit. And this remains as long as we have a good, healthy, functioning immune system. Now, since we're moving on to 11.2, which is how colonization can lead to disease. So what happens when this microbial antagonism isn't enough and this pathogen actually colonizes us and makes us sick? That's what 11.2 is about. There's quite a few terms that are in this section that I thought might be useful to just list out before we get started. I think flashcards would be a great idea with these terms, so you will practice those to know what they mean. A pathogen, hopefully we know that one by now. It's just an organism that can make us sick, basically. It's an organism that can infect a host and result in some kind of infection or disease. Now, there's two sub-classifications of pathogens, and that's true pathogens and opportunistic pathogens. True pathogens are capable of causing an infection even when a person is healthy. On the other hand, an opportunistic pathogen is seeking an opportunity to arise. It waits for when our immune system is compromised to be able to become established part of our body. So it's not normal for these opportunistic pathogens to actually make people sick even when they're exposed to them because they have a healthy immune system. But in other cases where that's not the case, where we don't have a healthy immune system, these opportunistic pathogens take over. So a few other definitions. Infection. This is when a microbe passes our host defenses and enters our body. A disease is any kind of deviation from normal health. And an infectious disease is specifically a pathological state that is caused by a microbe. So typically an infectious disease is oftentimes something that can be spread because it's caused by a microorganism, like staph infections of the skin or E. coli infections of the gut or the UTI tract. A disease, on the other hand, is not necessarily caused by just a microorganism. It can have other factors that typically are not necessarily going to be transmissible. Like, for example, diabetes is a disease, but it's not something that you can catch from another person. It's not necessarily caused by an infection of a specific microbe. So there's our difference in terminology there. And then finally, virulence and virulence factor. Virulence is the severity of a disease caused by a particular microbe. How severe of a disease can it cause? The higher the virulence, the easier it is to get into a host and cause damage. And a virulence factor is any characteristic or structure that increases or contributes to the virulence of any kind of organism. So we'll go into some examples of that in just a few minutes momentarily. But before we do, let's talk about some factors that can weaken our host defenses and make us more likely to be susceptible to an infection, and specifically those opportunistic infections. So age is a contributing factor. So older age, being elderly, or being very, very young are typically two factors that allow for us to become sick more easily. Genetic defects and immunity. and even acquired defects in immunity. So for example, any kind of genetic defect that would affect one of the many of our white blood cells, or any of the proteins that are part of our immune system, and enzymes. We'll get it all into our immune system in the next few chapters, but for those who have taken anatomy and physiology, you may have heard of some of them, like complement or interferon, and we'll get to others later. But AIDS, for example, that's an acquired defect that is caused by HIV, which is a virus. And what it does is it attacks our immune system. So again, we see an immunocompromisation that allows for more host susceptibility to infection of various microbes. Surgery, organ transplants, when you're healing, that's when your immune system is going to be compromised. If you have any underlying diseases, diabetes, liver malfunctions, cancers, you're going to be immunocompromised. Pregnancy, chemotherapy, any immunosuppressive drugs. For example, when you are on chemotherapy, typically part of that is going to be an immunosuppressive drug or if you've had a organ transplant, or a skin grafting, or if you have multiple sclerosis, or oftentimes one treatment for autoimmune diseases may be immunosuppressive drugs. So that's going to compromise the immune system and allow for illnesses to happen more readily compared to a healthy immune system. Pregnancy as well weakens your immune system. And there are lots of other examples, but other infections, if you have any co-infections, would additionally further weaken the immune system and allow it to be more susceptible to being infected with other bugs, other microbes. All right, so now let's talk about how colonization leads to disease. And there's multiple steps of this, and of course we'll start with step one. In step one, we're going to breach a portal of entry. And so to understand that, we need to know what a portal of entry is, and I think it's very intuitive. A portal of entry is a route that a microbe will take to enter the body. So, for example, if you have a cut on your skin, that could be a portal of entry. Your mouth consuming incidentally various microbes when eating food or drinking water or anything else, that is a portal of entry. Inhalation is a portal of entry. Even through the skin, some organisms can absorb through the skin. So those are just a few examples of portals of entry. And most pathogens have a very specific portal of entry. And I'll give you a great example that we probably are very familiar with, and that's the influenza virus. And as we probably are aware of, that causes the flu. And we're familiar with it probably because we know every year it comes around and comes and goes and comes and goes. Well, the way that specific virus colonizes the body is through the nasal mucosa. And the nasal mucosa is essentially the mucous membranes within the nose. So we inhale it, breathe it in, and that is what we call inoculation. Just like inoculating an agar plate like you've done in lab, it's where you're introducing a microbe to something. Here we're just talking about it in the sense of the human body. So we're inoculating the nasal cavity with this virus by inhaling it. Now, if we were to inoculate the skin with this influenza virus, then no infection would occur. It has a specific portal of entry it must enter in order to make us sick. Now, some pathogens, on the other hand, can have more than one entry point, and some examples are listed here, like Mycobacterium tuberculosis. You can probably guess that causes tuberculosis, but it can enter both through the respiratory tract and the gastrointestinal tract, so either breathing it in or consuming it. Streptococcus and Staphylococcus bacterium can enter through the skin, especially if there is a cut. It can enter through the urogenital tract, which is how urinary tract infections occur, and even the respiratory tract by breathing in aerosolized particles of it. So next I have a table that comes from your book that shows the various portals of entry and some organisms that are typically involved. Please take note of the upper right hand corner that's highlighted in yellow that you don't need to memorize this table for the exam. I'm just showing you examples. Now previously, the ones that I've went on and talked about, like the ones on the previous slide that I'm showing now, yeah, I would probably know those. Now I'm not going to guarantee you they'll be on the exam, but I can guarantee you that every single one of these would definitely not be on the exam. So don't memorize this table. This is not going to be asked of you on the test. I just want to show you some examples. So you can see the skin of as a portal of entry has various examples of organisms that can infiltrate it. We see the GI, the respiratory tract, and the urogenital tract, and various microbes that can enter through those portals of entry to cause disease. All right, so now let's move on to another term called infectious dose. And if you think of the word dose, I know a lot of y'all are probably interested in healthcare, so you're probably familiar with the word dose or dosage, and it's the amount of something, right? What's the dose that I'm supposed to take of ibuprofen if I have a headache? It's how much. And so in this case, the infectious dose is how many microbes are necessary to make me sick. It's the minimum number of microbes that is needed to cause an infection. And this number can vary depending on the microbe, because the virulence of microbes vary from one to another. And again, virulence is how easily the characteristics they have that make them more susceptible to cause infection. So for example, let's look at the very bottom of this list. We see cholera. Cholera is a disease caused by a bacteria called Vibrio cholera. There are a couple other strains of or species of Vibrio like Vibrio vulnificus, and there's two others. I can't remember them off the top of my head, but essentially for those to cause infection, we're going to need to be infected with, let's see here, 1 trillion cells is what I'm seeing. So that's a lot. That's a very high infectious dose. That means we have to have a lot of bacteria of this specific strain, which is typically going to be consumed in this case, that would be required to cause disease. On the other hand, we see typhoid fever only needs about 10,000 cells. Gonorrhea, 1,000 cells. TB, Giardia, coccidioidomycosis, only 10 cells. And then all the way at the top here, we have our most virulent organism, which causes Q fever, and only a single cell is needed to cause an organism to become ill from that bacterium. So, all in all, again, infectious dose is the number of microbes necessary to cause disease, and it varies from one organism to the next. So, now that we have entered into the portal of entry, let's talk about step 2, which is adhesion. So now that the microbe has entered, it has to adhere to a surface. And this is going to occur differently depending on the pathogen. So, for example, some pathogens are going to contain fimbriae, and hopefully you remember that term from previous chapters when we talked about prokaryotic organisms and their structures. There are those little bristle-like structures that are used as adhesive structures. They have an adhesive function. Sometimes surface proteins that are embedded in the cell membrane can act as proteins that will adhere to surfaces. Some bacteria may have slimes or capsules that make them sticky, and so it will cause adherence to surfaces. And by the way, when I say surfaces, I'm talking about the surfaces in your body. Just to clarify, I'm not necessarily talking about the table here. I'm talking about inside of you. Viruses, they have to attach to specialized receptors that are proteins that are embedded into specific cells within the body. And even parasites like our worms, helminths, they are going to adhere to our bodies, typically in the GI tract, and they're going to fasten themselves there with various structures. Some of them have little suckers that will suck on to the GI wall. Some use hooks, some use barbs. All in all, basically adhering to our bodies. So now, let's move into step 3. So step 1 was getting inside, portal of entry. Step 2 was adhering to some surface of the body. And step 3 is now we got to get past the host defenses. And so essentially for this, for us, it means that it's got to get past our immune system. So our immune system is made up of all different kinds of cells, and we'll get into that in much details in the later chapters, I believe in the next two. But one of the concepts or one of the cells that we'll talk about are phagocytes. And these phagocytes are cells that will engulf and destroy other cells, such as pathogens. And whenever they do that, they will have enzymes inside them that will destroy the pathogen by tearing it apart, to make things simple. However, some pathogens have what we call anti-phagocytic factors, which makes it hard for our white blood cells to act on them. And so one example of a anti-phagocytic factor is a leukocidin. And these leukocidins can be produced by bacteria that outright kill phagocytes. And if you need help remembering this term, break it down. That's what I always tell my biology and my A&P people that have taken A&P with me. Leuko means white, and anytime you see cide, it means to kill. So we have killing of white blood cells. And so in this case, these bacteria that produce leukocidins will kill phagocytes, which are an example of white blood cells. Next, the slime or capsule that we talked about that some organisms have will supply that bacteria with an additional surface layer that can make it difficult for that phagocyte to eat it or engulf that pathogen. And even more interestingly, some bacteria can be engulfed by a phagocyte and survive inside of it. Pretty cool. All right, in step 4, causing disease. So now that we have survived the host immune system, we will wreak havoc. And there is 3 ways that we will wreak havoc. And when I say wreak havoc, I mean cause damage to the host. So the first way is directly through enzymes or toxins. And those toxins can be classified as endo or exotoxins. So what we're seeing, and that's what we're seeing in this first picture, okay? So here we see the microbes are secreting enzymes. The little blue balls are the bacteria, and you see the enzymes in the little tiny little dots that are throughout. So these are giving you examples of bacteria cells that are excreting enzymes that are going to be toxic to these human cells. Another example is shown here where we see bacteria in blue. And again, we see all these little dots that are exotoxins, and they are going to cause damage to these healthy human tissues that exist here in this layer. So these are epithelial cells. For those of you who don't know, which would be maybe my biology students, but that's basically any cell that is going to cover a body surface, like the lining of the gut or your skin, even though I can tell these aren't skin cells because they're not stratified squamous, but that's a whole nother story we won't get into. Anyway, so our second way that our bacterial cells can cause disease is indirectly by inducing the host's defenses to respond excessively or inappropriately. And so that is shown in picture B here. So what we see is a phagocyte that is trying to consume a bacteria, but it can't be due to this capsule. So the bacteria cannot be engulfed. And so as a result, if this happens, let's say in the lungs, if the bacteria in the lungs have capsules and the immune system tries to pour fluids into the lungs, to try to get defensive cells there, we are trying to get more and more white blood cells here, that causes the symptoms of pneumonia because we have now fluid in the lungs. So this would be a response from our immune system that results in a negative consequence for the human body. And then number 3, when we have epigenetic changes made to the host cells by microbes. So in this scenario, bacteria will successfully infiltrate inside of a host cell, travel to the nucleus, as you can see here, and it will contain microbial proteins and enzymes that will be released into the nucleus that can then modify genetic material. So I don't really want to go into too much details on here, but what you see, CH3, For those of y'all that may have had chemistry, y'all may know that's a methyl group. But epigenetic changes is a change to DNA. And so in some cases, these microbes can produce these proteins and enzymes that can cause methylation. And that's going to alter the the genes that are turned on and off in a cell, which can then ultimately create damage to the host cell. Because, like I always say in both A&P and biology, you need the right concentration of the right proteins at the right time in the right spot. So if we have this bacteria that's now manipulating our genome and making us produce things or prevent us from producing things that we need, it will cause cell damage. All right, so let's move back to that topic of toxins. So that was from number 1. I just want to move back just to show you what I'm referring to. We're still talking about the same stuff. We're just going into more details. So in the first example of how we can cause disease, we said it was through toxins. So what are toxins and these enzymes? So some of them can be classified as exoenzymes. Notice I wrote this in red because they are enzymes in the extracellular fluid. So, meaning outside the cell. And so, if you were to look at body tissues, and hopefully we all know this from a prerequisite course, and I know my drawing's pretty crappy, but the round things are my cells, here's some kind of basal membrane, and there's space in between cells. And that space is called the extracellular matrix, or I call it the ECM for short. That's the space within cells. So exoenzymes are enzymes that can exist in this extracellular space, and they're secreted by these microbes. And these exoenzymes can break down and inflict damage on these cells. It can dissolve these the host's defense barriers to promote the spread of disease to other tissues. Let's say maybe hypothetically that this was skin. It has many layers. Technically the cells should be flat, but we're doing the best I can with the little drawy thing I have. But if it damages all these cells in this basal membrane, now it can get below the skin, lead to further infection like cellulitis. Not good. That's what exoenzymes are capable of. Some examples include the following. And I love these because typically they kind of, the wording fortunately is very intuitive. If you have mucinase, y'all see mucus. So ACE always means enzyme. So I know my biology people know that, not sure about the A&P. ACE equals enzyme. If you have an ose at the end of a word, it's typically a sugar. In this case, we have ACEs, ASE. So mucinase is a enzyme, because it ends in ACE, that digests the protective coating on mucous membranes. So some bacteria produce this enzyme to digest the protective coating that lines our mucous membranes of like our nose, our GI tract, the stomach, the intestines, etc. Hyaluronidase. This is another enzyme that digests the substance that cements animal cells together. Ladies, particularly, you may be familiar with hyaluronic acid that is oftenly found in facial care products. So this particular bacteria can digest that to make our skin more susceptible to further infection. Coagulase is an enzyme that causes coagulation, and coagulation is clotting. It causes clotting of the blood or plasma. And kinases, they are responsible for dissolving fibrin clots. So these are a very few number of examples of various enzymes that bacteria can possess that can affect the host in negative ways. Now, what about toxins? A toxin is a chemical product of a microbe that we consider a poison to other organisms, and they're typically named so according to their target, like neurotoxins acts on the nervous system, enterotoxins acts on the intestines, and later we'll talk about enteric bacteria, which, as you can then guess, is the bacteria in the intestines. Hemotoxins, hemo meaning blood, nephrotoxins, Nephro meaning kidneys, and for you AMP people, you should know that because y'all should be familiar with the kidney structure and nephrons. Biology people maybe not so much, but nephrotoxins are toxins that damage the kidney. So now let's move to exotoxins, which are excreted by pathogenic organisms. They can cause the production of certain exotoxins like hemolysins. Hemo meaning blood, and lysin coming from the word lyse, which means to break or rupture. So these are a class of exotoxins that can cause the destruction of blood cells, which is called hemolysis. And in fact, what we're seeing here in this picture is a blood auger. It's one of a way that we can try to go down a rabbit hole in identifying a bacteria. We'll get to that at the end in our fourth unit. But what we're seeing here is a blood auger plate. So it is an auger, just like we've talked about before, like in chapter 2 and other labs throughout our laboratory sessions. But this is sort of like a gelatinous substance, more like a really, really thick jello. So that jello-like, quote-unquote, substance has this nutrients in it that's going to be required for microbes to survive. So it's basically got all the stuff in there that makes microbes survive. But in addition to that, it also has sheep's blood, typically. Not always sheep's blood, but oftentimes it is sheep's blood. So what we're seeing here is they inoculated this blood auger plate with 3 different microbes, and one of which we would call this one beta hemolytic. And you don't need to know this yet, but beta hemolytic, meaning that we see complete hemolysis here. Here we see what we call alpha hemolysis. That's the symbol for alpha, where we see a little bit of hemolysis, but it's not complete. And here we see something called gamma hemolytic, and that means that it did not have hemolysis occur. So whatever bacteria this is clearly did not produce a hemolysin, but this one for certain definitely did, because they cause the red blood cells to hemolyze and release this hemoglobin pigment, which causes an empty space if there's complete hemolysis, and if there's alpha hemolysis, that we can still see that shaded. It almost looks kind of greenish in real life, because that alpha hemolytic bacterium burst the red blood cells and release that hemoglobin, which has a red pigment. But here, combined with the blood auger, it just looks a little green after hemolysis has occurred and it has been oxidized by the air. So now let's move to endotoxins. So notice the difference here. We're moving from exo to endo. Our endotoxins are are actually parts of the outer membrane of the cell wall, specifically in gram-negative cells. This is important. Oh my goodness, sorry. I can't stop it. There we go. Gram-negative cells. And so before we read all this stuff on the PowerPoint, let's just look down at the pictures. What we've been talking about prior was this. Exotoxins that, oops, I didn't mean to like scribble it out, but exotoxins that are excreted from a bacteria, this being our bacteria. Now we're moving into endotoxins. The endotoxins are these purple things, and as you can see, they're embedded into the outer membrane of this cell, and this cell is a gram-negative cell. Gram-negative. So on a gram stain, it would be pink. Just FYI, as we've seen before. So, what are these little purple rectangles actually? They are largely in the form of something we call lipopolysaccharides, or LPS. You need to know that. Lipopolysaccharide. Don't overcomplicate it. You can break it down. Lipo meaning it has a lipid component, and polysaccharide meaning many sugars. It's a combination of sugars and lipids. So these complexes together we call lipopolysaccharides, or LPS for short. So these LPSs have a variety of systemic effects on tissues and organs. Systemic meaning body-wide. They can cause fever, inflammation, hemorrhage, diarrhea. If we have blood infections by some of these, it can be particularly dangerous. And some examples include Salmonella, Shigella, Neisseria meningitidis, Escherichia coli. Those are just some that are particularly dangerous and can definitely lead to endotoxic shock and can be fatal. So Salmonella you've probably heard of because of things on the news, but it's typically associated as a GI bug with poultry. Shigella, another GI bug. Neisseria meningitidis causes meningitis. Escherichia coli can cause various things, but a lot of times it's associated with GI tract infections, but ultimately some strains can go into severe issues with shock and be potentially fatal. So now let's move to our next slide, which discusses the origins and effects of these circulating exo and endotoxins. So here it's kind of summarizing the things that we've discussed already. So you don't need to memorize this table per se, but it kind of just summarizes the things that we've talked about already. So exotoxins are classified here. They're in this column. We have endotoxins on the right, and then we talk about their toxicity and these various things in the center. So if we compare exotoxins to endotoxins, exotoxins are pretty toxic, very in minute amounts, whereas in endotoxins, we need high doses for it to be toxins. So the exotoxins were the things like we talked about, the endotoxins were the things that we talked about on that last slide. Exo was prior. So next, effects on the body. So we talked about fever and inflammation and various things. Again, you don't need to memorize all of this. Chemical composition, small proteins or enzymes was exotoxins, and here specifically we have lipopolysaccharides, which you do want to know that. So again, we have some variations in heat stability, etc. But again, you don't need to memorize this chart. What I do want you to know are the things that we went into detail about. Okay, number 2 and 3. We are now going to discuss inducing and damaging the host and how it responds, and then finally that epigenetic change. So moving back just a moment, let me make sure we're all oriented to the same place. Now we've talked about this one. Now we're going to talk about the other two in more detail. So 2 and 3. So inducing a damaging host response, and number 3, epigenetic changes. These two methods of cell damage are more recently discovered than the first one that we have discussed. In number 2, the host's excessive or inappropriate response to a microbe will cause damaging effects to the body, just like the pneumonia response we talked about initially, but there are others, like cytokine storms. So this was something I actually heard on the news a good bit with, what was it? It was some medication being taken with COVID-19, but I can't remember because it's been so long ago. But what this is when we have a overproduction of these molecules that we call inflammatory cytokines. These are normal in the body, and what they do is they help clear infections, but they also, as a byproduct of their actions, cause damage to tissues. So a cytokine storm is whenever we have these overproduction of these cytokines, which is going to wind up causing more damage than it is clearing infection, and thus ultimately having a negative impact on the host. And then again, we mentioned this one already, but it's when the bacteria in the lungs have capsules. And again, not all bacteria have capsules. Some do, some don't. It depends on the species. But if our immune system detects them, it's going to push fluid into the lungs to try to get more immune cells there, and that results in pneumonia. And then finally, number 3, our epigenetic changes is when a microbe is going to alter our host DNA. So again, we talked about that methylation event, which essentially is going to shut down or activate genes, turn them on or off, which will ultimately then change the proteins that we are producing. We'll either stop producing proteins that we should, or we'll start producing proteins that we shouldn't, and that's going to cause a negative impact to cells, because once again, cells have to have the right proteins and the right shape and the right time and the right concentration. And so when that gets out of whack, it can lead to disease. And sometimes microbes can cause this. So let's next talk about some patterns that we see in the medical community of infections. We're going to start with some terminology. There are some subtle differences here, but these are very, very important, and it's not going to leave you. I would be willing to bet at least some of my paycheck that you're going to see these and be expected to know the differences again when you get to further courses, especially if you're in nursing. So a sign, a symptom, and a syndrome. A sign is objective evidence of a disease noted by a clinician. A symptom is a subjective evidence of a disease as sensed by the patient. So what in the world does that mean? What's the difference there? Subjective is something that can vary from person to person. So for example, my pain tolerance is subjective. If I say, let's say you and I have the same, we both have a splinter in the same exact spot in our foot. To me, that pain can be a 7, but to you it may be a 2. Because pain is subjective. Everyone has different pain tolerances. And so this is why it's considered subjective. It's dependent upon the patient, and that's why it's also evidence that's presented by the patient. So for example, if I say I have a headache, that is a symptom. It's not a sign, because a clinician, which would be a doctor or nurse, etc., they cannot look at you and tell that you have a headache. So comparatively, a sign would be something that is measurable. All right, so objective is something that we can see. It's objective, it's measurable. For example, I could take your blood pressure. That is an objective piece of evidence. I could take your heart rate. I could take your temperature. Based on those signs, I could guess that you probably do have that symptom. But again, that's that subtle difference. Signs are noted by clinicians. It is objective evidence that we can measure somehow. And then symptoms are voiced by the patient as to how they feel, which is subjected to their individuality. A syndrome, on the other hand, is a disease that is identified by a certain complex of signs and symptoms, meaning that typically these things go together. So, for example, if if I have a patient that has a fever, that is a sign of something. If the patient says that they feel like they're having trouble breathing, that is a symptom because it's subjective. They're telling you that. Now, let's say I did an x-ray of the lungs and I see fluid in the lungs. That would be a sign. So, because we have a fever and we have a fluid in the lungs, we could consider that this person most likely has pneumonia, which would be a syndrome. Okay, so let's do another example. So symptoms of inflammation can be as follows: fever, pain, soreness, and swelling. So those are our symptoms of inflammation. If we have signs of inflammation, we have edema, which is the accumulation of fluid. We can physically see that, and it can be noted by a clinician. We can see abscesses or granulomas, which are walled-off collections of inflammatory cells or microbes. Lymphadenitis, which is the swelling of the lymph nodes. That's why a clinician will feel around on your neck to determine if they detect that. So, again, the symptoms are listed prior, which is the ones that are identified by the patient and are subjective. Signs by clinician. Now, I will say fever may be arguable depending on if temperature is actually taken. If you go to the doctor and say, I think I have a fever because I have a headache, then that would be considered a symptom. If I have pain, again, that's subjective. Soreness, that is subjective. Swelling is somewhat objective. Like if I say I have swelling of my legs, well, again, that's a patient announced thing. So in order for it to be considered a sign like edema, then the clinician would have to notate it. Let's look at a second example. So symptoms of a blood infection. Symptoms of a blood infection typically include fever, chills, abdominal pain, nausea, vomiting, diarrhea, and fatigue. But if we're looking for signs of a blood infection, meaning that a clinician is going to look for these indicators, they would identify things such as leukocytosis. And remember, we said leuko means white blood cells. Cyto means cells. And we have osis at the end that typically means a disease of or an increase of in this case. So we have an increase in white cells. If we have an infection, our immune system cranks up and we start producing more white blood cells. Now, sometimes we can also have, ironically, leukopenia, which is a decrease in the white blood cell levels, depending on the causative agent. We also have the term septicemia. This is a generic term which simply means that there are bacteria or some kind of pathogen that is in the blood and multiplying. Then we have bacteremia and viremia, and you can probably guess what these mean. Bacteremia means that bacteria is in the blood, and viremia is where we have viruses are in the blood. But both of these indicate that they're not multiplying, so that's that subtle difference again between septicemia and bacteremia and viremia. Septicemia is where they are present and multiplying, and not good, not good, not good. Okay, so now next, sometimes infections go unnoticed because there's not any noticeable symptoms, and the microbe can still be active in host tissues, but because they are not having any actual symptoms that they're feeling, the host may not seek medical attention. And these type of infections are called asymptomatic, meaning A, without symptoms, or subclinical, meaning it's not obvious, or it means it's inapparent. Alright, so let's now move to step 5. So we've gone through steps 1 through 4. Now step 5 is vacating the host. And to vacate the host, our microbe will have a portal of exit instead of a portal of entry. And this is the avenue by which the pathogen will leave the body. Some will shed through secretions, excretions, discharge, sloughed off tissues. It really depends on where they they're harbored in the body. There's a lot of microbes in these materials that increases the likelihood that the pathogen will reach other hosts, unfortunately. And a portal of exit is usually the same as the portal of entry, but some pathogens can use a different route. It just depends. So in summary, here were the steps that were involved that we've discussed when a microbe causes disease. We said that it first has to have a portal of entry, then it has to have a method of attachment, it has to survive the host defenses, it causes disease, and eventually exits the body. Alright, so next. Not all pathogens like to enter and then simply leave our body. Unfortunately, even after patients get better, there are some microbes that will still persist, and this is called latency. And you may remember this term from our viral chapter, which was, I think, chapter 5. But this period of latency is when a microbe can go into a dormant state and result in a chronic infectious disease. Viruses, or certain viruses anyway, are pretty famous for this because it's not necessarily unheard of for them to do this, like herpes, simplex viruses. Those are the ones that can cause genital warts, or some strains can simply cause fever blisters, cold sores. So those are viruses that can essentially flare up and then become unactivated for a while, and there'll be some triggers that can initiate that, and then it can go away again, and so on. The herpes zoster virus. This is the virus that causes shingles, which can occur later in life for individuals who contracted the chicken pox essentially earlier on in life. Hepatitis B is another example of a virus that can go latent. HIV, which results in AIDS as well, and Epstein-Barr virus. Some other organisms that are capable of latency are bacteria and protists, though. So, for example, syphilis is caused by a bacteria, and it can go on. It has like multiple stages, 4 stages that can start off from just typical illness signs and symptoms, like almost like the common cold or flu, that eventually get worse over a long period of time. Typhoid fever, tuberculosis. Tuberculosis can take a long time to kill the host, and malaria. So all of these are examples of organisms that can go through latent phases. So now, another thing that some organisms can do is that even after a patient may get better, there may be persistent issues, and these are called sequelae. They are long-term or permanent damage to tissues or organs that was caused by that initial infectious disease. For example, if you are infected with a microbe that causes meningitis, it can lead to deafness. So it is a long-term permanent issue that is basically a secondary issue that was caused by that microbe. So the meningitis caused the deafness and not necessarily the bacterium directly. With strep throat, obviously it causes aggravation and irritation of the throat, but it can also have sequelae that include rheumatic heart disease. Lyme disease can have sequelae, long-term effects of the tissues that can lead to arthritis. Polio can lead to paralysis. So those are all examples of sequelae, long-term issues that are a result of an infection. So now let's move on. Let's move on to the course of an infection, and I think this is really interesting personally. So it describes the time period from our initial exposure to the time that we are rid of the bug, whatever it is, virus or bacteria, etc. And so what you're about to see on the next several slides is this same picture, and you'll see that there's going to be certain areas highlighted that coincide with the phase that we're talking about. So at this point, it's showing you the initial exposure to a microbe at the beginning, and this is showing you over time, and then this graph is indicating the intensity of symptoms. So we can see initially at the incubation period, which is this part, we're going to have pretty much no symptoms. The agent that we're infected with has just entered the portal of entry, but there's not enough of them to really elicit enough damage to cause symptoms. And this time period can range from several hours to several years, and that I think is really interesting. And let me give you a great example for those of y'all who may have children, but if your kid brings home a stomach bug, you will get it, and you will get it that night, because their incubation period is very, very short, and I can tell you that from firsthand experience when some of the kids have brought home their lovely stomach bugs. Typically probably going to be a viral infection in that case. The majority of infections, though, typically range from 2 to 30 days, despite the fact that the actual range for all bugs can be very, very wide, from several hours to years. So that's our incubation period. No disease or no symptoms yet, but it's entered the portal of entry. Next, we have our prodromal stage. This is typically the first 2 days after the incubation period. We start to have mild discomfort symptoms. We just don't feel very good. You might have some headaches, muscle aches, fatigue, upset stomach, and just general malaise. You just feel like poopy. Then, as we can see in our next phase, the acute phase, this is the height of the infection. This is when the infection agent has multiplied to a point where there's lots of them that are in the body. This is the time period where it will have its greatest effect, its greatest influence on our target, which is us, or on its target, which is us, our target tissues. This is where the pathogen is at peak activity, and we typically are going to see fever and very prominent specific signs and symptoms depending on the pathogen that we're infected with. And this length of time can be variable. It really depends on the microbe. So next, we're going to have our convalescent period. This is when the patient begins to respond to infection. Either, like our white blood cells and our immune system, etc., take over, and it starts to get rid of this pathogen that has infected us. So we can see a decline in symptoms. And this is the period of time that I think it's relevant for healthcare people to know, when many patients may stop taking antibiotics that were prescribed to them. And I think it's important for y'all to know that, because that's not good. It's always important for your patients to take all of their antibiotics that they have been prescribed at the times that they have been prescribed to take them, because even though they're feeling better at this point in the convalescent period, they still have pathogens in their system. And so if we stop taking antibiotics, let's say right here, well, the bugs that are left in our system are left over because they're a little bit more resistant to that antibiotic than the ones that have already been killed off. So now guess what? We stop taking that antibiotics, and the ones that are left in our system are a little bit more resistant to that antibiotic than the other ones, and they're going to proliferate and make more bacteria or whatever the bug is that are more resistant to that antibiotic, just like they are. So this is one of the leading reasons and mechanisms behind antibiotic resistance. So very important to explain to your patients to always take every single antibiotic that they are prescribed when they are prescribed it. Moving on to our last phase, continuation phase. This is where we are going to have the organism might linger for a little while, and this only happens in some infections. Sometimes the lingering can be months, years, or even indefinitely, even if the patient feels fine. Or the organism is gone, but symptoms can continue. Just depends, again, on the organism. All right, finally moving into section 3. We're going to now talk about transmission. So how do we contract these bugs? How do they get into our portals of entries? Where do they come from? Well, to start with, we're going to need to define a few things, and that is reservoir, carrier, and transmitter. So if we think of like a reservoir just as like a generic English term, hopefully you're thinking like, a place where something's held. Like a water reservoir would be a place where you would hold a basin of water, I suppose. So a reservoir, when it comes to microbiology, is the natural primary habitat where a pathogen typically resides. Sometimes reservoirs are living things. So humans can be natural reservoirs for certain microbes. Many other animals can also be natural reservoirs for certain microbes. Non-living reservoirs, on the other hand, of course, are non-living, such as soil, water, and air. And our other definition that is under reservoir here, carrier, is an individual who inconspicuously shelters a pathogen. And what that means is that they happen to be very lucky, and they are harboring a bug that is not making them sick, but they can pass it to other people and make them sick. For example, let me just check real quick. I want to make sure I didn't have it in this PowerPoint. For example, you can look this up, but Typhoid Mary. Typhoid Mary had typhus, and it didn't make her sick. She was a carrier, but she infected many people during the course of her lifetime because she was asymptomatic. A transmitter is an individual or object from which an infection is acquired. So it's Basically, in this scenario, typhoid Mary was a transmitter of typhus. So these terms can sometimes be interrelated for that reason. So, like, for example, syphilis is a reservoir, but it's also a transmitter when it comes to humans. So humans are the reservoir and we can transmit it. Hepatitis A, humans are a reservoir for it. We also can transmit it via contaminated food. So sometimes these terms can kind of overlap. Now, when we have animals that serve as reservoirs for infections, we call these zoonoses. Now, typically when we talk about zoonoses, we're not referring to humans. We're talking about other things like other mammals, birds, lizards, even smaller things like arthropods, fleas, mosquitoes, flies, ticks, mites. I mean, the list can go on and on. But these are examples of organisms that can cause zoonotic infections, and they are infections that are indigenous to animals but can naturally be transmitted to humans. So again, the majority of reservoir agents are arthropods. That's important. Ooh. And especially around here, mosquitoes. We know mosquitoes in South Georgia. But other things, fleas, for example, back in I don't know the years, but the Black Plague, you might have heard it before, called the bubonic plague. Mosquitoes, West Nile virus, Zika virus, certain types of flies like tsetse flies, which is predominantly in Africa, can cause African sleeping sickness. Ticks are definitely a source of infection for various microbes. Let's see, like Lyme disease, which is caused by a bacteria. Now, also, there are larger animals that can spread infection as listed. Some mammals can spread rabies, raccoons, dogs. Birds can also serve as a method of spreading zoonoses, such as, I have a hard time with this one, pisacosis, there we go, and lizards and other organisms that are related can harbor salmonella and cause salmonellosis. I cannot say that. Salmonellosis. There we go. But obviously that's related to the salmonella bacteria. Okay, so zoonoses make up a full 70% of all new emerging diseases worldwide. And I thought that was interesting to know because that is a very, very high number. Now, next, here are some common zoonotic infections, and note at the top right hand corner, again, you do not need to memorize all this, just showing some examples. So we already mentioned rabies. Yellow fever can be caused by birds, mammals, mosquitoes. Influenza, we can get it from chickens, birds, swine, hence the avian bird flu, and then we had the swine virus, the one from pigs before. West Nile virus can be harbored and transmitted from mosquitoes, and et cetera, et cetera, et cetera. Again, you don't need to know this list. Know the ones that I talked about in detail. So now let's look at our next slide. So agents of infection that can be spread from one host to the next are said to cause communicable diseases, and they are contagious. So think of it like in the community, communicable community. It is a disease in the community, so we call it communicable, and it's contagious from one person to the next. Some diseases are more contagious than others. For example, influenza, the flu virus, is very, very, very contagious, meaning it's very communicable. The measles is another example. So whenever we see a measles outbreak, everyone panics because we know, as in the medical community, how quickly it can spread. Hansen's disease, on the other hand, is caused by a bacteria that results in the condition leprosy, and it's very weakly communicable. So it doesn't really spread as easily from one person to the other. Some examples of non-communicable diseases are listed below. Cancer, diabetes, those are things that aren't necessarily associated with microbes. Tetanus, on the other hand, is, but it is also not typically spread from one person to the other. And a lot of times, many mycoses, and if we recall, that is infections caused by yeasts and molds, basically fungi, are not very easily, are not all communicable. All right, so now let's talk about the patterns of transmissions that we see in these communicable diseases. They can be either horizontal or vertical transmission. So I like to think of this like a family tree. So if you have your parents, that's your mom, that's your dad, this line is showing that they have children, and here's you, and here is your brother. Now, if you took my biology class, you may know that I should have drawn the boys as squares, but we're not going that deep. So mom, dad, their children. Horizontal transmission is where we have spread from one infected individual to another via various contact, whether it be direct, indirect, or a vector transmission. We'll get back to that in a second. On the other hand, vertical transmission is when we have a disease that's transmitted from parent to offspring in specific ways via ovum, meaning the egg, sperm, placenta, or milk. So meaning essentially prior to birth or through the process of breastfeeding. That's what vertical transmission is. So you can think of it like this. It's the parents transmitting the disease to the children. Now, can we also have horizontal transmission that goes from parents to children? Yes, but it's not going to be through ovum, sperm, placenta, or milk. It will be through close contact. For example, when I told you my kid brought home the stomach bug, that would be horizontal transmission, even though it's my child, because it's not a transmission that occurred in utero or through breast milk. Indirect transmission is where we have an object in between. Basically, if I were to touch a object like the counter, and let's say I am sick and have a contagious infection, and I touch the counter and walk away, and then another person behind me comes and touches that counter in the same spot and then touches their face. Say it's like the flu virus. So now they've been infected with the flu. That indirect transmission, that object, the counter in this case, is considered a fomite. A fomite is any inanimate object that can harbor some kind of bug due to someone coming in contact with it that is sick, and then someone who is not sick also coming in contact with it later, picking up that infectious agent. Another example of indirect transmission is transmission through a vehicle, and when I say vehicle in this case, I'm not talking about your car. I am talking about some kind of natural, non-living material like air, water, food, or soil. So things that are natural, versus the fomite being more like an inanimate man-made object, like here's a good example: baby toys. Oh my goodness, or kids' toys. We all know kids are gross. Okay, so if they're sick and they've got a runny nose, they're going to be playing with these toys, and then they're going to walk away and play with something else, and another little kid's going to come behind them and play with that toy that they just put their snotty hands on. So that would be an example of a fomite, whereas something more natural that is not man-made would be a vehicle. And then lastly, number 3, we have vector transmission. This is where we will have an arthropod that will harbor an infectious agent and transfer it to a human. And again, arthropods are things like ticks, mosquitoes, fleas, flies, etc. Now, another very important thing for healthcare workers to be familiar with is healthcare associated infections. They are also called nosocomial infections. And these infections are diseases that are acquired during a hospital stay. So they acquired this disease or infection while they were at the hospital. They got it there. So they call them, for this reason, not healthcare associated infections because it was contracted in a healthcare setting. And there's anywhere from 0.1 to 20% of all admitted patients with an average of 4% that contract nosocomial infections when they enter and are admitted to a hospital. So that is something of very much importance to our healthcare related field workers. There are 750,000 cases a year, and 75,000 deaths that are a result of these healthcare acquired infections. So that's a very big deal for healthcare workers. And I know you might be thinking, wait. Aren't hospitals supposed to be the place where we treat infections and not get them? Well, yes, but think about it. Hospitals are places where people go when they're sick. So hospitals are going to be full of immunocompromised patients. And then these immunocompromised patients have infections, and even patients that are not immunocompromised may have some infections that can then pass them to immunocompromised patients. So basically, hospitals are like it's like Mardi Gras for pathogens. It's like a field day for them. Any kind of clinical setting is going to be a field day for pathogens because it is a place where sick people congregate, and then they touch surfaces, they utilize medical equipment, they touch other patients, they go to the bathroom, they touch personnel, they have visitors, and so on and so forth. So it's just kind of a cesspool when it comes to pathogens. Isn't that lovely? So it's very important that healthcare workers are familiar with it and proper procedures are put in place to eliminate the possible spread of nosocomial infections as possible. So let's look at the 5 most common ones. And y'all, I'm sure we'll hear these again in later classes. The most common hospital-acquired infection is Clostridioides difficile. Oftentimes it is abbreviated as C. diff. C. diff for short, and it causes GI infections which can lead to dehydration and potentially death. Staphylococcus aureus, it can cause pneumonia, it can cause skin infections, particularly in clinical settings at surgical sites, and can transmit to the bloodstream and cause septicemia, which is definitely not good because it can be fatal. Klebsiella species. So Klebsiella are an enteric gram-negative rod, and these can cause GI or urinary tract infections. They can also result in pneumonia. It can also infect surgical sites and wreak havoc there. So it's kind of a very variable bug that can cause multiple issues, and therefore, unsurprisingly, it thrives in hospital settings where there are people that are having surgery, that have UTIs, that are having pneumonial issues, and so on. The next one, E. coli, is a urinary tract infection causative agent typically, but it can also cause surgical site infections, and it can also cause septicemia. And finally, Enterococcus species, multiple different species, but they as well can cause UTIs, bloodstream infections that can lead to septicemia, and surgical site infections. All right, so remember those for the future, not just for my class, because you will see them again. I would be willing to bet, and if you don't see them again, I would be shocked. So next, and I think this is our last section, is 11.4, which discusses epidemiology. And this is the study of the frequency and distribution of infectious agents and disease and other health-related factors in a population. And a population means, essentially for us, mostly in people. So us as a human race would be considered the population. There are diseases that government agencies have determined are reportable, or also called notifiable diseases. And so there are organizations that certain diseases, if a patient has it and a clinician diagnoses it, then it must be reported to these authorities. Other diseases can be reported on a voluntary basis. It really depends on the rules, regulations, laws that are at work in the various agencies that can be local, district, state, national, or even international, that keeps track of these infectious diseases and their numbers. So some examples would include your health department. So every county in every state has a health department. The CDC, this is the Center of Disease Control. The health department reports to the CDC. And the CDC is the principal tracking organization for the United States. The CDC reports to the WHO, there we go, the World Health Organization. So it's kind of like a chain of command almost, where we find numbers in the local areas, report it to the state level, we report it to the United States level, and then to our WHO, which is the international level for the countries that participate. So the outcome of these studies that the public health departments and the other affiliated organizations come up with is going to help us to develop prevention mechanisms and treatment programs, and develop a basis for predictions on what we think may become a cause for concern presently or in the future. So here's a list of reportable diseases in the United States. Again, note, you don't need to know all of these. You don't need to know this list. But as you can see, some of them are pretty gnarly bugs. So things like the causative agent of anthrax, which is Bacillus anthracus. We have West Nile disease, Western equine encephalitis, which is caused by a prion, very, very dangerous, and can't kill it, unfortunately. Let's see, botulism, which is caused by a bacterium, brucellosis, also bacteria, campylobacteriosis bacterium. These right here, these carbamazepine producing organisms, they are resistant to certain antibiotics, which is why they are reportable. Because again, like we've mentioned, antibiotic resistance is a huge deal in the medical community, and we really try to track these very well so we can prevent as much as we can further conditioning of bacteria to our treatment methods. Chlamydia trachomatis causes chlamydia. We've talked about cholera. Let's see, cryptosporidiosis, dengue fever. That's also caused by a bacteria, I believe. Diphtheria is a bacteria. Many of these are. HIV is a virus, so is Hep A, B, and C. So various organisms can cause these diseases, but these are the ones that are reportable to the U.S. So if a patient has this, they're required to report to their the health department's required, or let's say you just go to a doctor, not necessarily the health department. Your doctor will report it to the health department. The health department reports it to the CDC, and the CDC will report it to the WHO. Here are some other ones. Again, you do not need to memorize this list, so we'll move straight forward. So what do they do with this information now that they have it at the state level or the national level or the international level, etc.? They're going to use this data to determine some things such as prevalence and incidence. And prevalence is the total number of existing cases in a population. How prevalent is it that we have this infection? And so to calculate that, they'll add up the total number of cases in a population divided by the number of people in the population. That'll give them a decimal number, and so if they multiply that by 100, it will give them a percentage. So for example, the prevalence of smoking among adults in the U.S. is currently 14%. And so how they gathered that data is to take the number of people that smoke in the population, divide it by all of the people in the population, and they would have got 0.14. To convert it to a percentage, they would multiply by 100 to get 14%. The incidence is the number of new cases over a certain period of time. So to calculate incidence, they take the number of new cases in a designated period of time, and they divide it by the total number of susceptible people. So for example, the incidence of new tuberculosis cases in the United States in 2018, because remember we said it's in a designated period of time, was 2.8 people for every 100,000 people. And it's not uncommon where we utilize the units of per 100,000 people, to give us a realistic idea of how common it is. So 2.8 per 100,000 people. Okay, so let's look at some example of some data that's been collected by epidemiologists. So our first graph up here at the top shows us our number of measles cases that have been reported since 2010 up to 2019. And we can see the number of cases is on the y-axis and the time periods are on the x-axis. So we see relatively low numbers. We see a nice little yikes spike here in 2014 and a very yikes spike in 2019. So would this be considered a prevalence or incidence? This would be considered an incidence report because it's over time. It is over a period of time from 2010 to, from in the year 2011 there's data 12, 13, 14 all the way to 19. And then looking at picture B, this is showing us the number of lung injury cases that are reported to the CDC as of November 19th, 2013. So in this case, it shows it in a color-coded fashion, where the fewest cases, their states would be in that light pink. In fact, I'm not sure. Alaska. And then as we move down in the color codedness, we'll see more and more cases, with the highest being in green in California, for example, over there, Texas, etc. All right, so these statistics are very important for epidemiologists, because it helps them to establish the rates of disease, meaning how often they occur, with regard to certain physical attributes of the population, such as sex, race, geographic region that they live in, etc. They are often followed seasonally, yearly, and even long term, so that mortality rates, when morbidity rates can be measured. Mortality rates are the number of deaths in a population due to a certain disease. So the overall death rate from infectious diseases over time has dropped. However, the number of persons afflicted with infections, which is called the morbidity rate, has remained relatively high. Note that difference between mortality and morbidity. The morbidity rate is just how the number of persons afflicted with the infection, how many get infected. Mortality is how many die from the affliction. So here's another couple of examples of epidemiological data. We have a example of data that shows a common source epidemic in our first picture, and it results from common exposure to a single source of infection over a period of time. Another name for these are point source epidemics, and essentially it's because the infection has a common source, a single source, and so everyone's infected at once. Let's say that it is at, Lord forbid, Splash in the Borough, and there is a infection that is spreading in the water there. Well, what we're going to see in the population are large bumps, where when people go to that source, we see a high infection. Then it goes down, high infection. Whoop, there's summertime, high infection. We have periods of low, and then we have highs and lows and highs and lows and highs and lows. So it's this dramatic increase and decrease of the number of persons that have this illness over time. On the other hand, we have propagated epidemics, which result from an infectious disease that is communicable from person to person. So in this case, since the source is transmissible from person to person, we're going to see a sustained long-term hump in the graph, if you will. So instead of having consistent up and down, up and downs, up and downs, because it's communicable, again, it's transmissible. I caught it at Arby's or wherever, and I spread it, and then someone else spread it to someone else, and so on and so forth. This is what we see with COVID-19. So we see this huge increase and eventually a downswing of the data. Some additional epidemiology terms that you should be familiar with are as follows. The index case. This is the first case that brought the epidemic on it that was at the attention of the official. So it may not necessarily be the first case, but it's the first one that was diagnosed. Endemic is an infectious disease that exhibits a relatively steady frequency over a long period of time, specifically in a particular geographic location. So, for example, malaria is endemic to certain parts of Africa because it is relatively common there all the time. We don't see it very often in America. Sporadic means it is a case where we have occasional cases reported that are irregular. An epidemic is when statistics indicate that the prevalence of an endemic or sporadic disease is increasing beyond what is expected. So initially, for example, we would consider we considered at one time COVID-19 to be an epidemic. However, eventually we started calling it a pandemic, pan meaning everywhere, because it spread across continents. So epidemics are typically going to be things that don't cross the continental barriers. Now, here's something you might be thinking, well then, is the flu considered a pandemic or an epidemic? I'm sure scientists argue that, but technically I would say no, because it's something we expect. We know that the flu is going to spread through our population at certain times of the year. Now, if it was beyond what was expected, like the Spanish influenza of 1918, yes, that would be an epidemic and probably actually a pandemic, because I'm pretty sure that one went cross continents. And that is the end of our lecture for chapter 11. I think this one's probably the longest one, so at least you have for this semester of spring 2023 anyway, spring break to sift through it if you want extra time. But I hope it's helpful, and I will get back with you on our platform in Blackboard soon. Have a good week.